To introduce our speaker, Colt received his bachelor's degree in biomedical science from Northern Arizona University. He is currently a College of Biosciences 2022 master's candidate at Kansas City University, and his current ambition is to attend medical school in order to become a physician. So with that, the floor is yours.
My name is Colt Solberg. I work in Dr. Robert White's lab on the Duchenne Muscular Dystrophy project, and the title of my presentation today is Identification of the Human Retinal Dystrophin Promoter, a Potential Pharmaceutical Target for Duchenne Muscular Dystrophy. And as was stated earlier, Dr. Olson gave a fantastic presentation on this. However, our modality for treatment that we're attempting is a little bit different, and you'll see that here in a minute. So just to give a quick background on Duchenne Muscular Dystrophy, Duchenne Muscular Dystrophy, abbreviated DMD, is an X-linked recessive genetic disorder resulting in progressive muscle degeneration disease. It results in the absence of dystrophin protein in the muscle, and this absence of dystrophin inevitably leads to the disease state. Patients who have DMD lose the ability to ambulate by age 12, generally, and then generally die prematurely by age 20 to 30 due to cardiomyopathy or respiratory issues. Currently, there is no real cure for Duchenne muscular dystrophy. The only modalities we can do is focus on improving patients' quality of life through the use of corticosteroids or through assistive devices. And there are very promising gene therapy treatments that are being implemented, such as CRISPR, as we've heard about, exon skipping and other modalities. However, the biggest issue that we run into is T-cell mediated immune responses with these types of treatments. However, our lab may have discovered a novel treatment utilizing an isoform of dystrophin known as retinal dystrophin. And this is an endogenous protein that occurs in approximately 30% of the patients that have DMD. So before we start talking about retinal dystrophin, let me introduce the dystrophin protein. So as you can see here on the left-hand side, the dystrophin protein has 4 functional domains. So we have an N-terminus that has an actin binding site, a series of spectrin-like repeating domains, as well as a cysteine-rich domain and a C-terminus that binds to the syntrophin of what's known as the dystrophin-associated protein complex. And really what this does is, if you think of a building, it's like a scaffold for a building. It keeps that cytoskeleton of the sarcolemma really compact and prevents it from falling apart. Now, when we look at the gene for dystrophin, the dystrophin gene has several different isoforms. We have a cortical form, muscle, Purkinje, as well as retinal dystrophin, which is the one we're interested in, followed by Schwann and glial. Now, each of these isoforms have their own unique first exon as well as their own unique promoter. When we look at retinal dystrophin as the protein, we actually note that retinal dystrophin, much like full-length dystrophin down here, has an N-terminus with actin binding site, which you can actually see here in the image, the second actin binding site found in the spectrin-like repeat of full-length dystrophin. It also has the spectrin-like repeating domains, a cysteine-rich domain, and a C-terminal domain. So functionally, these are equivalent functional domains to full-length dystrophin, which gives us the thought, what if we could induce this protein in skeletal muscle? Would it be able to compensate for the lack of dystrophin in these patients? And in doing so, our lab had developed a human retinal dystrophin transgene that was implemented into mouse embryo and then put into pseudopregnant female mice to produce transgenic mice that produces retinal dystrophin in the skeletal muscle tissue. And again, as we stated beforehand, so retinal dystrophin, we'll go back one more slide, is constituted by its own first unique exon, exon R1, and then exons 30 through 79. So this transgene uses those exon R1, intron R1, and then exons 30 through 79. So again, can inducing retinal dystrophin in the skeletal muscle tissue of a DMD model mouse or DMD patients, would this be able to rescue them from their disease state? Well, if you look here, the image on the left here actually is 2 litter mates. They are brothers. And so these are mice that have the mouse on the left-hand side is a DMD model mouse that has the transgene implemented in skeletal muscle tissue. And the mouse on the right-hand side is a mouse that has the DMD, is a DMD model mouse, but does not have the transgene implemented. And just gross observation, you can obviously see that there is a distinct difference between their 2 states. As well as when we look at the lifespan curve of our DMD model mice that have the transgene, these mice actually live a similar lifespan to that of our Black 6 control mice, whereas our DMD model mice end up dying anywhere from 3 to 5 months of age. We also noted that there's a rescue from the scoliosis, or in mice, it's kyphosis of these patients. So this is a mouse, DMD transgenic mouse, that has a normal curvature of the spine, whereas the DMD model mice without the transgene still exhibit the abnormal curvature of the spine. And then using immunocytochemistry, we're able to actually show that retinal dystrophin attaches on the sarcolemma to the same area as full-length dystrophin. So we're able to actually show that, again, to us, this is could be used as a compensatory mechanism for treating Duchenne muscular dystrophy. So really what we noted in all of our studies of these mice is that the expression of retinal dystrophin in DMD model mice changes a severe lethal myopathy into a very mild and viable myopathy. And this leads to my portion of the project. The goal of my project is actually to identify the promoter for retinal dystrophin so that we can identify drugs or biological compounds that we can use to upregulate retinal dystrophin in the skeletal muscle tissue for DMD patients. In doing so, we're actually going to be utilizing several different DNA regions that we have selected using different bioinformatic programs, and I'll show you that here in a second. And we're going to be implementing those DNA regions that we believe may contain the promoter. We're going to clone those into a luciferase expression vector plasmid, and then transiently transfect those into a cell line in order to test a luciferase assay to determine which of these does in fact produce luciferase, and that would show us that this is the DP260 promoter, so the retinal dystrophin promoter. And here is our diagram of the 3 different DNA promoters that we're looking at. So these nomenclatures, the FARI1, 2, and 3, these are in-lab nomenclatures. These are nomenclatures that we gave to these. So FARI1 is indicated by a promoter program known as Genocopia. FARI2 is a promoter program known as Promoter 2.0, and then FARI3 is a promoter program known as Softberry. And what's unique about each of these promoter regions or potential promoter DNA clones is that each of them has transcriptional elements or promoter elements. So we have cat and tata boxes in each one of these regions. Now, as we said beforehand, we're going to be transiently transfecting these luciferase plasmids into an appropriate cell line. And this appropriate cell line for us was the WIRI-RB cell line. The reason that we selected it is because it's a retina-derived cell line. It's a retinoblastoma cell line. And really, the reason we selected it is because we believe that it would already be producing or have the mRNA for retinal dystrophin production. However, when we looked through literature, we did not find anything that actually showed this. So we needed to demonstrate this ourselves. So using the primers that we had selected, we were looking for a 400 base per product. And in fact, we did get that using RT-PCR on the cell line. However, to be good scientists, you can't just say, okay, I found a 400 base per product, this must be it. So we needed to identify that this truly was retinal dystrophin. And in doing so, we used chromatograms and then blast analysis to identify that, in fact, yes, we have a 100% match rate. So this is, in fact, retinal dystrophin. And in fact, it was RT-PCR performed because we don't have an intron present anymore. So really what this data showed for us is that the WIRI-RB cell line was an appropriate cell line for our transfections of the luciferase plasmids. Now, moving forward with the transfections, we're using a lipofectamine 3000 as our transfection media. And in order to test for transfection conditions in these cell lines, we're actually using a GFP plasmid. So this is a green fluorescent protein plasmid. And what we're trying to do is, again, determine what conditions are going to be best for transfection for our cells. And in our first set of experiments, we were able to actually get some transfection, as you can see here. However, these are superimposable images, meaning that this image, both these images are the same, just one is under different conditions to show the GFP. As you can see, not all of these cells are transfected. And in actuality, our transfection efficiency was only 10%, as shown by flow cytometry. So currently, we're still in the process of determining what are the best or optimal conditions for transfection using lipofectamine 3000 into our cell line. And then for our future goals for the project, really what we're trying to do is, once I've identified the transfection efficiencies, we're going to move forward with transfecting the transient transfection of the WIRI-RB cell line, or of the luciferase plasmids into the WIRI-RB cell line to show which plasmid does in fact give us luciferase activity with the luciferase assay. Once we've identified that DNA promoter, we're then going to move forward and stably transfect a human embryonic kidney cell line with that plasmid that gave us the expression. And the reason we're doing this is that the human embryonic kidney cell line, much like muscle cells, should not produce retinal dystrophin in it. And again, the long-term range of this goal is we're trying to upregulate retinal dystrophin in muscle tissue, in skeletal muscle tissue in DMD patients. Once we have stably transfected the human embryonic kidney cell line, we're then going to submit it for high throughput screening of drugs and biological compounds, so that way we can see what drugs can we utilize to upregulate production of retinal dystrophin in the skeletal muscle tissue for these patients. And in doing so, once we've identified those drugs, we're then going to move forward and start treating our DMD model mice with these drugs. And hopefully we can get the same results that we've noticed with the transgene. Once we've identified that, we'll be able to look at different modalities for treatment using that drug, such as, at what age group do we need to start treating at, what is the dosage, so on and so forth. And again, I'd like to reiterate that one of the most important aspects of this project is that because retinal dystrophin is endogenous in 30% of the patients, we would not have a T-cell mediated immune response. So these long-term immunosuppressions that are required in a lot of the the gene therapies that are seen, such as CRISPR, we don't have to immunosuppress our patients. They'd be able to just take the drug and hopefully start to, progressively get better. As acknowledgements, I'd like to thank Dr. Robert White for all of his mentorship and for his help in the lab. I'd like to thank Bradley Thornton as my colleague who's helped me a lot in the lab, as well as Alec Graff and Keenan Swan, who have been big proponents to the lab, as well as Amber Wiggins McDaniels, who's really been the mastermind behind teaching me all my lab techniques. Without her, I don't think I'd be able to even pick up a pipetter. So,
thank you, Colt. Nice job. Questions from here in Kansas City? Dr. Zaidi.
Excellent presentation. Thank you so much. I have a question regarding that GFP data that you showed,
yeah.
and I may have missed that, but was the transfection efficient? Is this only GFP or is it GFP fused to the retinal dystrophin?
So no, this is only a GFP plasmid. Again, we're at this point just trying to determine the transfection efficiency. What we actually are going to do in the future is a co-transfection with both the GFP plasmid as well as our luciferase plasmid.
Any other questions from Joplin?
I'll ask a question. This is Dr. Staudinger. Two questions, really. In your mouse model, do they have any cardiac defects? MCK is a muscle-specific promoter, a skeletal muscle-specific promoter. And Duchenne muscular dystrophy patients, while they have skeletal muscle defects, eventually die of cardiac defects, heart failure. So are you planning on using a heart-specific promoter? So that's two questions. Do the mice have a cardiac defect, and how do you envision delivering your transgene to cardiac muscle in the future?
So I guess I think there's a slight misunderstanding on that. The transgene was to initially show us the effects of retinal dystrophin in the skeletal muscle tissue of the mice. It uses a muscle creatine kinase promoter and enhancer, but our goal actually isn't to utilize a transgene in the patients for treatment, it's to utilize a drug therapy to target the promoter that we've identified to then upregulate retinal dystrophin in skeletal muscle tissue. But as far as what you're saying, we did in fact see cardiomyopathy in our DMD model mice. And we did actually note that the DMD model mice with the transgene did rescue from cardiomyopathy.
Then a follow-up question would be, let's say you do identify a drug that upregulates retinal dystrophin. So I think I understand now what you're saying, and that is, if the drug distributes to every tissue in the body, then it would upregulate that promoter in cardiac as well. Am I understanding?
Correct.
Okay, thank you.